All right, so the next lab that you need to do, if you look on the Canvas module, is called the Discovery Lab. This one's really quite simple. It's actually meant for you to have a little fun with it. If you look at it when you open it up, basically, I just want you to go outside, get some sun on your skin, and basically take 5 pictures of 5 animals, 5 pictures of 5 plants. Even if you don't know what the hell the plant is, what the hell the animal is, take a picture of it, and then next to it you'll see a bunch of lines. I just want you to describe it in your own words. In other words, discover kind of the world around you. It could be your backyard. It could be if there's a pond by your house. If you want to go to a park with the social distancing stuff, feel free. Go right ahead. So 5 animals, 5 plants. Easy enough, all right? So now the next lab that I need to talk about is one of the, what we now, I would call, quote, the real labs, whereas if we were meeting face to face, you would then have microscope labs. So this would all be microscopic type of stuff. So the first lab is going to be the prokaryote lab. Specifically, it's going to be paying homage to cyanobacteria. Why? First off, cyanobacteria are a lot larger than what we call normal bacteria, like pathogenic bacteria. They're really tiny. But they're easier to see, they're easier to label along with that. But one of the reasons why I have you do an homage to cyanobacteria is because they're one of the major contributors to the Earth's atmosphere of oxygen. Basically, in the early primordial soup, you're looking at the oxygen levels on planet Earth were less than 1%. Currently, they're 21%. The cyanobacteria are the ones who pretty much almost single-handedly brought it up to that so that you can go, you can breathe. So you're saying, by doing the lab, you're saying, thank you for, giving me the oxygen to breathe, okay? Because they're one of the major contributors to it. Now, so if you look at the lab on Canvas, then it starts off with, again, prokaryote, okay? Now, it's not going to look like this because I took the time to actually get photos, print the photos in the lab. I have them in order that I'm going to show you, and then I have a box next to them on the file on Canvas that tells you specifically what I want you to label on each picture. Okay, so understand they are prokaryote. Prokaryote, they're not going to have a nucleus. You're not going to see the nucleoid. It's way too small. It's way too thin. You're not going to see the bacterial ribosomes. They're there, but you can't see them. You'll be able to see cytoplasm, you'll be able to see a cell wall, you'll be able to see a cell membrane. In a lot of cases, you can see a gelatinous capsule around it. The capsule looks like a clear bubble area around it is what it looks like. It almost looks like when you would think of like a cell wall. That's kind of what it looks like. Please, for the love of anything good and pure, do not label in nucleus because it's not on my list. It's not going to be on the list. They are prokaryote. Pro means before, karyote means nucleus. These are creatures before there was the evolution of the nice little nucleus that was formed, membrane-bound nucleus. All right, so to kick it off then, if you look, first creature I have you look at is a creature called Anabina. Anabina, depending on how you want to say it. So, Anabina. All right, this one. Now, when you do a lot of bacterial work, like if and when you go into microbiology, ways to classify bacteria would be like, are they solitary? Do they live in groups? If they live in groups, how does the group form? Do they occur in a strip? Then it's called strepto. Do they occur in a bunch? That's called staphylo. What are the shape of the cells? Are they round? That's a coccus. Are they rod-shaped, like a Good and Plenty or like a Mike and Ike type of thing, a hot tamale, those candies? That's called bacillus. If they're a corkscrew, that's called spirillum. You can also then, if they cause a disease, whom do they infect? How do they infect? What do they go after? That's another way to classify. New ways they use to classify now are basically genetic, DNA markers as well as RNA, protein, like you did for phylogenetics lab, protein similarities between certain aspects, the amount of guanine, cytosine content. There's a whole list, whole genome, you name it. That's more the modern viewpoint of it now. But if you ever go to microbiology, what you'll have to do is you'll have to classify unknowns. And how you're going to classify them is like I'm showing you. What's the shape of the cell? How do they grow in a group? In a strip? Do they grow in a clump? Do they grow in certain types of media? Are they aerobic or are they anaerobic? All these are different things that you would utilize in a micro lab to figure out an unknown. Now think, in a medical lab, that's not as practical because it takes time to get the culture, to grow the culture out. Meanwhile, the person's still sick, and it may 3, 4, 5 days for a diagnosis, and you finally figure out, oh, it's this bug. That doesn't help the person who was sick the last 3 to 5 days. So there needs to be a faster way to diagnose, which is what you do when you go to like a clinic or a lab nowadays. Now, anyway, none of these are pathogenic, understand. Anabena. Anabena is going to look like a strip of little balls. Every so often, some of the balls are going to be bigger than other ones. Okay? Now, so one way to call that, you could say, well, that is streptococcus, a strip of balls. Usually when we do cyanobacteria, we don't use the term bacillus, coccus, and spirillum in most cases. Usually we just call them filamentous, meaning they're in a long chain and they look like little filaments. But it will help you whenever you go to study them or describe them, look at the shapes of the cells because it will distinguish one from the other. Now, the most common type of anabena you'll see will be small little balls that are grainy. Those are called vegetative cells, meaning a run-of-the-mill cell. These are the ones that are photosynthetic. These are the ones that produce oxygen. Every so often, you'll see a bigger cell, and it will be empty. It looks like it's clear. That is going to be called a heterocyst. Heterocyst is a strange little structure that you see in some types of cyanobacteria. This allows the bacterium to do something called nitrogen fixation. The majority of the atmosphere surrounding you that you breathe in, over 70% of it, 75% of it, is looking at nitrogen gas. Your body doesn't want it. It can't use it. Nitrogen, however, is a major component, hopefully you know, of fertilizers, nitrate. Well, very few things on planet Earth can take atmospheric nitrogen, pluck it like it's an apple off of a tree, and make it into nitrate, a plant fertilizer. The heterocyst is what does that for this bacterium. Then you'll see another ball, which is, you know, the big ball, but it won't be empty. It'll kind of look grainy, but it'll be a bigger ball. What is that? That is called an endospore. Endospore is the equivalent of an escape capsule. If the environment goes badly for the bacterium, it can wall up its nucleoid, its DNA, into one cell, and therefore when the environment goes bad, the rest of the cells die, but now you have an escape capsule with which to wait for better times. You go into hibernation. So that's an endospore. Now, not all of these will make endospores, and the chance, quite frankly, of you seeing them in the pictures or in a lab is kind of low, because normally endospores are only made whenever you're under environmental duress, and when they make these slides commercially, the cells are happy and healthy. They're good, solid environments for them, so usually very few to little to no endospores. All right, so next one you see there is gliotrechia. Gliotrechia, if you ever watched, spelling is, if you ever saw the Lorax, think of like truffula trees, okay? That big fuzzball. So when you're on low power, gliotrechia kind of looks like a dandelion puff, okay? Well, understand each one of these threads coming out is a separate colony. They all converge on a major point. Now, if I were to take one of these and break it off, what you would see are a bunch of cells that look like rods laying end to end, but at the end, in the middle, there'll be a ball. So it kind of almost looks like a push pin, or think of like a cane, like a bamboo cane almost, with a ball on top. That ball is going to be the heterocyst. Those are vegetative cells. Again, you'd have the cell wall, and then you'd have the cytoplasm. On the inside of the cell wall, you would have then the cell membrane, okay? Notice no capsule. If it had a capsule, it would look like it would have a bubble around it, okay? All right, next one up, let's see, is Merismopedia. Merismopedia, whenever you see it, kind of think of like an Eggo waffle. Think of like a graham cracker, okay? So usually you have these colonies, which are rectangular, and then you'll have these cells that occur in pairs. Well, they don't occur in pairs, they actually occur in fours. But whenever you have the focus of the microscope, you can only get one plane. If you change the focus, you would see the two underneath. So there's two on top of two, all right? But it kind of looks like a cracker, okay? So therefore, you would see cytoplasm, cell wall type of thing. The area that you're in, that's a capsule, okay? This big, huge cracker part. It's a communal capsule. All right, next, oscillatoria. Ciliatura looks like vacuum hose. Okay, so basically, instead of now having these rod-shaped bacteria laying end to end like boxcars in a railroad, let's lay them horizontally next to each other like that. So it kind of looks like, you know, the corrugated vacuum hose is what it looks like. Each one of these is a separate cell. So again, you have cell wall cytoplasm. One of the more interesting traits about Ciliatura is every so often you'll get this crescent moon curve. That crescent moon curve is either going to be called a fragmentation or separation disc. That's whenever the colony gets to a certain area, it can break off and go another direction. All right, notice again, no capsule. Cytonemia. Cytonemia, again, looks like, again, bacillus. So what you'll have are these little tiny rod-shaped cells. But in this case now, what you'll see is this clear area around them, like that. That clear area around them, that if you think it was a plant cell, it'd be a cell wall, that's a capsule. Okay? So therefore, cytonemia, is, when it was first discovered, was said to have, quote, angel wings. The angel wings are the capsule on either side. So it kind of looks like the body of a butterfly with two wings. First person described it said angel wings, and the name stuck. Now, cytonemia can also have branches. Branches means that actually you would have a literally branch coming off of it. If it's a false branch, it means that it kind of makes a nub and falls underneath. And that's when you'll see also in the lab as well too. All right, Arthrospira is the next one up. Arthrospira, if you can't guess the shape of it, spira, spiral. It's a spirillum. So therefore, it kind of looks like a lazy helix. Okay? All right, next one, Lingba. Can't spell today. L-I-N, Lingba. Now, Lingba looks just like a Ciliatura, except for it's going to have a capsule. So again, you're going to have like these cells that look like this, but in this case now you have a capsule. Okay, so it looks like the corrugated hose type of thing. All right, next one is Nostoc. Nostoc is disgusting. Looks like whenever you see it in a lab, like somebody goes on your slide, and then you smear it around and look at it, okay? So basically when you see Nostoc, usually it's on a smear on a slide, so under low power, it kind of looks like a donut, and it has that characteristic pale yellow color that kind of looks like dry mucus, because that's essentially what it is. If you were to zoom in, say, to this area here, what you would see are small little chains of balls in this mucus sheath. That mucus sheath is the communal capsule. Notice you see short chains of balls. People go, oh, well, that's like Anabina. Notice, no endospore. There's no heterocyst. It's just small little short chains of balls. Okay? Next one, the last one on it is Rivularia. Well, Rivularia, I'm going to draw it again. Rivularia looks just almost pretty much identical to Cytonemia. This is the one that's going to have the true branching. And I believe, if I remember right, you had one more that I put in, which I think was brain fart. I can't think. I gotta look forward. Oh, that would be, yeah, Spirillum. Okay, so Spirulina. Spirulina, if you follow anything in health or anything like NASA, Spirulina is like the superfood. People, when you go to like a health bar, you want a shot of Spirulina in it. It's a blue-green algae. It's not an algae, it's a bacterium, but it's a Brisnum or blue-green algae. You want to see Spirulina, basically, you have a freshwater fish tank, let it go without cleaning it every so often. You see those little threads that form in it, more than like that Spirulina. So, Spirulina, understand, Spirulina, spiral. Unlike Arthrospira, Arthrospira was a lazy helix. Spirulina is a really, really tight curlicue instead. Okay, but Spirulina is one of the, quote, superfoods, really. You can basically have your entire nutritional diet just from Spirulina sheets if you wanted to. And I'm trying to think if that was everything. Give me one quick sec to jump forward. And at the end of it, there's going to be an article that's going to be talking about a different type of bacterium. All I want you to do is read the article, and then in the article, I want you to summarize why we don't care for it very much in our waterways. And that's the prokaryote lab. I'll see you next time when we go over the protostry lab.